So hi everyone, can you hear me?
Yep.
Okay, let me turn my video on too. Um, Great, so I'm going to be talking about dimension reduction and clustering. Just move this here. Okay, with IMG's dataset clustering tool, and this is a feature of IMG that we released about a year and a half ago. And so I'm going to be giving an overview of the different dimension reduction methods that we offer, and my apologies to those of you who may already be well versed in this material. So why are we doing this? Well, a lot of the ideas that went into this feature, especially the motivation and also the choice of methods to include, are very much informed by this Frontiers in Bioinformatics review article from 2022 by Rob Knight and others. And the first author there is George Armstrong, in case anyone can't read that. And this feature is meant to be used for high-level data exploration, sort of as a first pass of taking a look at your microbiome data that you've just sequenced. And if you've taken samples from different environments or along some gradients, such as different depths in the ocean, you want to have some kind of validation that each sample is genetically similar to other samples from similar environments and genetically different from other samples from different environments. And this tool may also reveal groupings in your data that you weren't aware of that can be explored further. And there's also a kind of quality control element to it. For example, you could use it to find out if you have any outliers among your samples. Microbiome data is inherently high dimensional and complex, and with dimension reduction methods, we can simplify the data set while hopefully preserving key biological information, and we can enable visualization and reveal patterns. And as a side note to that, enabling visualization is a very common goal of dimension reduction in all contexts, not just microbiome analysis. And most dimension reduction methods are able to reduce to an arbitrary number of dimensions that's lower than the original number of dimensions. But quite often the goal is to reduce the data to 2 or 3 dimensions so that it can be easily visualized. And once you've done the dimension reduction, applying clustering methods such as HDB scan, which is the clustering method that we offer, can provide some additional validation that there are meaningful genetic differences between the groups. So this is actually an update of a previous IMG feature, which was called genome clustering. And as I'm going to talk about now, we consider this to be a big improvement over that older feature for many reasons. First of all, while the older feature operated only on genomes in the genome cart or selected from available categories of genomes or metagenomes, the new feature can operate on genomes from the genome cart, genome sets or scaffold sets from the workspace, or even mixed sets from analysis data groups. In addition, while the old version had less customization of plots, in the new version, the plots are customizable and interactive, thanks to Anna's work. And the available dimension reductions that we offered in the previous version were PCA, PCOA, and NMDS, and we've kept all of those. And in addition, we also offer the more modern nonlinear methods tsne and UMAP. In the old version, clustering was not integrated with dimension reduction in a graphical way. Instead, the user was only provided with a dendrogram view of the clustering results. And in the new version, you can visualize the clusters produced by HDB scan directly on the reduced dimension space that results from the dimension reduction algorithm you selected. Finally, the code for the statistical analysis in the old version was written in R, and in the new version, we've rewritten the code in Python using scikit-learn, among other packages. So here's the setup page for running a dataset clustering job in IMG. And as you can see at the top here, we have an area where you can select the data sets you want to include in the analysis. And you can see that I've added 2 of my genome sets there since they show up in the box on the top right. And at the bottom, you can select the parameters that you want to apply to the run. And on the bottom left, where it says clustering type, that's basically where you're choosing what kind of input data you're going to be using. You can select one of the functions like cog, pfam, or ko, or you can choose to produce input that's based on taxonomy or on one of the function categories like cog pathways. Once you've selected that, you can select a dimensionality reduction method to use in the middle column below. And then you can select what the dimensionality of the output of that method should be. So do you want to look at your data in 2D or 3D? Currently, there's only one choice for the clustering method that's going to be applied after the dimensions have been reduced, and that's the HDB scan algorithm that I mentioned earlier. So I'll go into more detail about the individual dimension reduction and clustering methods soon, but first I want to show an example of what the input data matrix looks like. And as you can see, I've selected PFAM here on the left, on the bottom left. So this is a small excerpt of the input data matrix that would result with the setup that I selected in the previous screenshot. And this screenshot comes from IMG's abundance profiles feature, and that's what we're looking at. It's an abundance matrix. And bear in mind that this is actually transposed from what the standard setup for running a statistical method like this would look like. We've got our metagenomes or individual observations in the columns, and we've got PFAMs, which are our dimensions or features in the rows. And I think there are something like 22,000 or so PFAMs, and there's a total of 65 metagenomes among the two genome sets that I added to the analysis. So this matrix is actually a lot bigger than just what we're looking at here. So this abundance matrix has an integer in each cell, and that integer represents how many hits each of the metagenomes across the top had to each of the PFAMs down the left side. And you can see that there are quite a few zeros, which means that there were no hits to that PFAM for that specific metagenome. And as a slight correction to that, I should note that this is actually an adjusted abundance matrix. You can see I selected estimated gene copies here under compute using instead of gene count. What the estimated gene copies option does, and it only works for metagenomes for which we have coverage information in IMG, is that each of the counts in the cells of this matrix have actually been multiplied by the average coverage of the corresponding contig, and this provides a way of accounting for different population abundances in the community. So let's move on to the dimension reduction methods. I'm going to talk about those now. The first is principal components analysis, or PCA, and this is the linear method that was first introduced by Carl Pearson in 1901. In PCA, each dimension of the reduced space, which is also called a principal component, is a linear combination of the original features and explains or accounts for a certain amount of the variance in the original data. So the first principal component accounts for the largest amount of the variance, and the second principal component accounts for the largest amount of the variance that's left over after removing the effect of the first principal component, and so on. So you're pretty much using linear algebra to rotate and rescale all of your data points in the same way into a smaller number of dimensions. The second method is principal coordinates analysis, or PCOA, which is also known as classical multidimensional scaling, or classical MDS. And this is similar to PCA, but more flexible. PCA can actually be thought of as a special case of PCOA. PCOA operates on a distance matrix of pairwise distances between the data points. And you can choose your own distance metric to build that matrix. If you use the standard Euclidean distance, then that would be equivalent to PCA. In our implementation, we use the Bray-Curtis dissimilarity. Other distance metrics that are sometimes used include the Manhattan distance and the Jacquard similarity. The goal of PCOA is to choose the configuration of points in the resulting low-dimensional space, such that the Euclidean distances between them best match the actual values of the pairwise distances in the distance matrix. And we're going to see how this constraint changes when we talk about non-metric multidimensional scaling. So for PCOA, while the operations on the distance matrix are linear, the distance metric used to generate the distance matrix may be nonlinear, which is the case for the Bray-Curtis metric that we use. So in those cases, you possibly have a little bit of sensitivity to any nonlinearities in your input data. So what makes a method linear versus nonlinear? Typically, the linear methods like PCA rely on linear transformations like rotating or rescaling. They preserve linear relationships, so if there's a straight line in the original data, that will remain a straight line in the reduced dimension space. And they tend to optimize for global properties like variance. The nonlinear methods, on the other hand, typically can apply different transformations to different areas of the data, bending or warping the data points differently in different areas. They typically focus on local structure and preserve local neighborhoods, and they can capture nonlinear structure in the data, such as curved manifolds. So now I'm going to talk about the three nonlinear dimension reduction methods that are offered in our feature, and the first of those is non-metric multidimensional scaling, or NMDS. For this method, you usually start with a randomly assigned configuration of data points on the low dimensional space, and then that gets iteratively refined. And the constraint that determines the refinement is that you need to preserve the rank order of the distances in the distance matrix, not the actual distances. In other words, the algorithm cares about which points are closer to each other, but not by how much. That's also where the non-metric part of the name comes from, since you're not paying attention to the actual values of the distance metric. This looser constraint in comparison to pCoA, which is the classical or metric version of MDS, makes it a more flexible and nonlinear algorithm that allows bending and warping of local areas. Another advantage of NMDS is that it can be applied to ordinal data, by which I mean ordered categorical data like ratings out of 5 stars. Finally, due to the non-metric ordinal characteristic, any outliers in your data will have a lower impact than they would for pCoA or pCA, similarly to the way in which the median is more robust to outliers than the mean. So I've been saying a lot of words about these dimension reduction methods, and now I want to show a few pictures in order to introduce the last two algorithms, tsne and UMAP. And what you see here is an image I got from Wikipedia that shows examples of handwritten digits that are part of the MNIST database, and that stands for Modified National Institute of Standards and Technology Database. And they collected and imaged a set of 60,000 digits, 0 through 9, that were handwritten by either American Census Bureau employees or by high school students. Each image of a digit is 28 pixels high by 28 pixels wide, and each pixel stores a number representing the grayscale value at that pixel. So the input data matrix has 60,000 rows for the digits and 784 dimensions or columns for the pixels. And when the tsne paper came out in 2008, they showed that their algorithm did a much better job than any previous dimension reduction algorithm on this data, in that it resulted in much better separation between the clusters representing each of the digits 0 through 9. So much so that in the paper, they write about how they even got separation between the regular sevens versus the continental sevens, which are written with an extra strike-through line. And here's an example of what those look like. So tsne stands for T-distributed stochastic neighbor embedding. And the way this algorithm works is similar to NMDS in that it uses iterative refinement of an initial configuration of the data points in the low dimensional space. tsne calculates probability distributions that represent the similarities between points. In the original high dimensional space, it uses Gaussian kernels with adaptively chosen variance or bandwidth. And in the low dimensional space, it uses t-distributions. The goal of the iterative refinement is to minimize what's called the Kullback-Leibler divergence between the two probability distributions. And that's just a way of quantifying the difference between two probability distributions. And tsne has several tuning parameters. In particular, the so-called perplexity affects how the bandwidths for the Gaussian kernels for the high dimensional probability distribution are chosen. So I should note that for both NMDS and for tsne, we're using a sort of meta algorithm where the algorithm is run multiple times, and then we choose the run that resulted in the lowest value of the loss that's being minimized. So for NMDS, it's the normalized stress, and for tsne, it's the Kullback-Leibler divergence. And the reason that this is the recommended way to do this for NMDS is due to the randomly chosen initial configuration of points. And for tsne, it's also due to randomness involved in the calculations that are part of the iterative refinement. So the final dimension reduction method that we offer is called Uniform Manifold Approximation and Projection, or UMAP. And UMAP came out in 2018. And it has similar visualization performance to tsne. And the theoretical basis for the algorithm comes from Riemannian geometry and algebraic topology. It's supposed to have better performance and therefore be scalable to larger data sets, and also is possibly better at preserving the global structure of the data in comparison to tsne, which can focus too much on local structure at the expense of any global structure. Moving on to the clustering method, and as I mentioned, we only provide one method, which is called HDBScan. And that stands for hierarchical density-based spatial clustering of applications with noise. And it's an extension of the DBScan algorithm. It was first published in 2013. and it has several advantages over other available clustering methods. So unlike k-means, which is another popular clustering algorithm, it doesn't require pre-specifying the number of clusters. Also, it can identify clusters with varying density within the same data set. Finally, it can handle outliers or noise well, since certain data points may be left unclustered if the algorithm determines that they don't fit well with any of the other data points. So let's go back now and take a closer look at those two metagenome sets that I added to the analysis when I showed the screenshot at the beginning of the talk. And thanks, by the way, to Simon for setting up this example. Like I said, there are a total of 65 metagenomes, and I'm adding them to the analysis as two separate sets. The first set has 28 epipelagic metagenomes, which means that these come from samples from the uppermost layer of the ocean, more specifically from the North Sea, not too far from Hamburg, Germany. And they were collected as a time series, which means that they were collected at different times over a certain time span from a single location and depth. And the second set has 37 freshwater metagenomes, which come from samples taken from lakes that are mainly in North America. And each sample was taken from a geographically distinct location. And about half the samples are from the lake water, and the other half are taken from lake sediment. So I ran all of the different dimension reduction methods on this data, and the one that resulted in the best separation between the groups was UMAP. So here are the results using the UMAP method, and you can see that there's a little drop down right here where it says color nodes by, and I've selected to color the nodes by cluster. So what you're seeing here is the result of running HDBScan on top of the UMAP dimension reduction results. And it looks like HDBScan found 3 clusters in this case with the different colors. And if we color the nodes by set name, I choose set name here instead of by cluster, we can see that the cluster on the top left corresponds very well to the epipelagic ocean water samples. And then there's these 2 other clusters, which both come pretty much exclusively from the lake samples. So to investigate this further, we can use the pointing device to draw a little rectangle around one of the 2 lake clusters, and then click on the View Selections button up at the top. And then we get this nice table that shows the information for the metagenomes that we selected. And by looking at the habitat column, and that actually needs to be added to the table using the metadata selector at the bottom of the table view, we can see that all of these metagenomes are from, or most are from freshwater sediment. And if we were to do the same thing with the cluster at the top right, we would see that those are almost exclusively from the lake water. So in this case, the dimension reduction and clustering tells a nice story, which is that we do indeed have meaningful genetic differences between the different sampling environments. And if you want to find out more about the differences between your groups, one possible next step would be to use IMG statistical analysis tool to find out which specific features, such as functional genes or taxons, are differently enriched in the 2 groups, in the groups. So here are some tips and tricks if you want to get started using this feature. First of all, there's no one dimension reduction method that always works best, so we provide the option of running all of the methods as part of a single run so that you can compare the results. And this should be considered to be for exploratory use. And if you're running any statistical tests on top of these results, you should definitely bear in mind that the validity of any p-values is based on the assumption that you've specified your entire analysis pipeline in advance. Also, it's a good idea to add your data sets to the analysis as genome sets, since that will allow you to color the points in the results graph by set. If you add all of your genomes from the genome cart, then all of the points will be colored the same in the color by set view. And finally, we provide a download bundle that contains all of the artifacts from the run, including the Python code and some graphs in PDF format. It also includes the full abundance matrix, which is the input to the statistical analysis. So if you want to run your own additional analysis outside of IMG, you can download the bundle and do that. So here's a list of potential improvements and new features that we're considering adding to this at some point in the future. First of all, we're planning to make some improvements in dataset clustering and across IMG that would allow the user to choose among various pre-processing steps, like applying some kind of log ratio to account better for the compositionality of the data. We're also considering adding Leiden-alg as an additional clustering algorithm to choose from. And we may add the ability to color points and plots based on certain categorical metadata. And finally, we may add an advanced features option where the user could enter different values of the tuning parameters of the different methods for different runs. And I'd like to thank all the people who worked with me on this project. Amy provided project management, Natalia and Reka provided the specifications. Anna worked on most of the UI elements, including the interactive plots. Ken worked on the generation of the input data sets and the integration of the Python scripts into IMG's existing computational infrastructure. And I worked on the Python scripts that carry out the different methods. And on this final slide, I've included a link to our user guide for this feature, as well as some references for the various methods. And thank you very much.